All right, greetings everyone, and thanks for coming to theme 6 of the user meeting. I hope everyone enjoyed the last short talk from Erin Nuccio, and now we'll move on to our next talk from Sabeeha Merchant. So we are extremely fortunate today to have Sabeeha talking. She is a professor of plant and microbial biology at UC Berkeley and also a faculty scientist in the Environmental Genomics and Systems Biology Division at Berkeley Lab. She is a very long-time JGI collaborator who originally got her bachelor's and PhD at the University of Wisconsin-Madison, and then did a postdoc at Harvard before joining the faculty at UCLA for many years before she moved to UCB relatively recently. She has earned numerous awards and accolades, which I won't enumerate, but those include election to the National Academy of Sciences, perhaps most notably. Her group focuses on algal biology, particularly the evolution of photosynthesis and the metabolism of trace metals. And she was an ideal speaker for this session because her group applies a variety of cutting-edge genomic and imaging technologies to these questions in algal biology. And so what we're going to hear about today is a collaboration with the JGI on the topic of widespread polycistronic gene expression in green algae. So welcome, Sabeeha, and we're looking forward to hearing your talk.
Thank you, Susanna, and thank you so much for giving me the opportunity to tell everyone about our recent discoveries. And in fact, I wish I could actually address all of the technologies that we have been using in my laboratory. So as Susanna mentioned, my lab focuses on the green algae, but I'd like to first point out that algae themselves are phylogenetically diverse in the sense that they have multiple independent origins. So each of these colored lines on this tree of life shows you branches that contain examples of various algae. The ones that my group focuses on and that we're interested in are the ones that belong to the Archaeplastida or result from the primary endosymbiotic event that gave rise both to the green algae as well as the green plants. So among the various algae that my group is presently working on, of course, is our favorite Chlamydomonas that I've been working on for, I guess over 30 years now, and was also Steve Mayfield's favorite, as you heard in his lecture. And then more recently, we've branched out, no pun intended, to work with Denaliella species, with Chromachloris ophingensis in a collaborative project with several investigators, I think some of whom are in the audience. And then most recently, when Jeffrey Mosley joined the group to bring in Oxygenchlorella protothecoides. So although they're all in the green lineage, they are easily, between 300 to 600 million years of evolution diverged from each other. And yet, as you will see from this lecture, there are aspects of gene expression that are conserved. And that's what I'd like to talk to you about today. When we were sequencing and assembling the Chromachloris ophingensis genome a few years ago on a DOE-funded project, Sean Gallaher, pictured here, discovered from the iso-seq data generated by Jane Grimwood and Jeremy Schmutz that many of the open reading frames in the Chromachloris genome appeared to be encoded on a single mRNA. And as shown here, light blue indicates one open reading frame, and then dark blue indicates another open reading frame. And it appeared from the iso-seq data that a single transcript covered both of these open reading frames. And that was a bit of a surprise to us at the time, because there was the dogma, certainly not textbooks, that this might be the case in prokaryotes, but typically in eukaryotic genomes, a single protein was encoded by a single mRNA. And in fact, the gene prediction algorithms use that, that dogma in order to define the longest open reading frame in a transcript. So this shows you the result from Chromochlorus zophingiensis. In the meantime, of course, Jeremy Schmutz had also been generating isoseq data for Chlamydomonas reinhardti, and I'm going to show you the same result also in Chlamydomonas, where you can see here on the left-hand side a light green open reading frame, and on the right-hand side here a dark green open reading frame. And again, a single transcript shown here covered both open reading frames. Now, reading the literature at this point, we noted that such polycistronic mRNAs had also been found in mushroom-forming fungi by Igor Grigoriev's group, and also from, I think, a group in China in cotton, where polycistronic again were transcripts again were discovered using the isoseq platform. But in those cases, those transcripts were not shown to be exclusively polycistronic, which I'm going to show you, nor were they shown to actually produce proteins, whether in vivo or in vitro. So what I'm going to go through now is some data analysis to document for you the fact that these mRNAs are that we discovered by isoseq were not representative of an artifact, for example, from PCR or from sequencing, but that these are genuine polycistronic mRNAs. And what I'm showing you is data from hundreds of transcripts found both in Chlamydomonas and Chromochlorus zophingiensis. So the first thing that we took advantage of was some data that Daniela Strenkert had generated through, I guess, a project that was, I think, over 5 years old at the time, where she had been looking at chromatin marks on the Chlamydomonas genome. And here the question was, if these mRNAs were genuinely polycistronic, we should find a promoter mark, namely the H3K4 trimethyl lysine mark, only at the promoter of the most upstream open reading frame. So we looked at the entire genome of Chlamydomonas at over about 100 such polycistronic mRNAs and asked the question, where were the promoter marks? If we look at monocistronic messages, we can identify these promoter marks upstream of the open reading frame. And then if we look at the polycistronic mRNAs, you notice that these promoter marks are found only on the upstream open reading frame and not on the downstream one. So consistent with polycistronic mRNA production. What about the other side of the transcript? So in this case, we would expect that the polyadenylation signal, in the case of the algae, the signal is TGTAA. We expect it to be found only associated with the downstream ORF, and likewise the poly-A tail only associated with the downstream ORF. So I'm showing you data both from the Chlamydomonas genome as well as from Chromochlorus zophingiensis. And again, what you notice here is that, of course, we see polyadenylation signals on all monocistronic transcripts. But then when we look at the polycistronic ones, we notice first that we see the signal only associated with the downstream ORF, indicated by the bar on the right-hand side here. And then when we look at the position of the tail, again, for both Chlamydomonas and Chromochlorus zophingiensis, we find the poly-A tail associated only with the downstream ORF. Again, consistent with a true polycistronic mRNA. The next question is, if these mRNAs were truly polycistronic, we could ask the question whether the two open reading frames were always co-expressed in hundreds of RNA-seq experiments. So we looked at that for monocistronic messages versus polycistronic messages. So the idea here is that you look at two open reading frames that are next to each other in the genome for all, in the case of Chlamydomonas, all 17,000 monocistronic proteins, and ask about the correlation between the expression patterns in various RNA-seq experiments. And of course, you find that for monocistronic mRNAs, these patterns are not at all correlated. That is, two genes next to each other are not necessarily co-expressed when we look at hundreds of transcriptomic experiments, but for the polycistronic ones in both Chlamydomonas as well as Chromachloris, we found that the two open reading frames were always co-expressed. Again, consistent with the idea that both open reading frames are encoded by a single transcript. All right, so the next question we had was to look at the proteins encoded by these open reading frames. And the question we're asking here is, are these the two proteins indicated in this case by the light blue and the dark blue open reading frame in Chromachloris ophingiensis, are the two proteins independently translated, or have we sort of missed a splicing event and mislabeled these transcripts as being polycistronic when perhaps in reality the open reading frames are fused? So the way we addressed this question was taking advantage of huge proteomic databases that were available to us through ENSEL and FICUS projects at PNNL, where Sam Purvine helped us to identify peptides corresponding to each of the open reading frames of interest among these polycistronic mRNAs. And what you can see, again, this is just an example of at one locus, you can see that, first of all, we identified peptides from both open reading frames. And most importantly, we identified actually the most C-terminal peptide in the upstream ORF and the most N-terminal peptide from the downstream ORF, confirming that the two proteins were actually independently translated. And when we look at this at a genome-wide level, we can confirm this, in fact, for most of these of the open reading frames that are encoded at these polycistronic loci. All right, so I showed you that the proteins are produced in vivo from polycistronic mRNAs, and the next question we had was, can we recapitulate the phenomenon of polycistronic transcripts being translated? Can we reproduce that in vitro? And in this case, I'm showing you the results from sort of 6 essentially random dicistronic messages that Sean chose. And they're listed here, the product shown in lanes 1 through 6. And what I'm showing you is the molecular weight of the upstream ORF and then the molecular weight of the downstream ORF. We translated them in either wheat germ extract or rabbit reticulocyte lysate. So of course, these are heterologous systems. And the results shown here is from the wheat germ extract system. And I'll ask you to focus on these arrowheads that show you the predicted size of the downstream ORF, the filled arrowheads, and then the predicted size of the upstream ORF. And you can see in each case for 6 of these, we were able to see the products from both the upstream and the downstream ORF, confirming that 2 proteins could be synthesized in vitro in this system. So the next question we had, because of course, as you heard from Steve Mayfield's talk, there's a lot of interest in using algae for synthetic biology applications. So the next question we had is, could we actually use this system in order to adjust the proportion of protein produced from the upstream and downstream ORF? And of course, we were also interested in the mechanism by which 2 proteins could be produced from a single mRNA. So what Sean did was he took advantage of the Kozak sequence that is associated with the initiator methionine. So here's an example of an endogenous sequence upstream of the initiator methionine. And he converted it into a stronger Kozak sequence, as well as a weaker Kozak sequence. And then he primed the in vitro translation reaction with these 3 different transcripts. So the endogenous one is shown in lane 1, where we get a certain ratio of the downstream to the upstream ORF. If he used a stronger Kozak sequence that would promote, oops, let me go back here. He noticed that the upstream ORF was better translated, whereas the downstream ORF was more weakly translated. And then if he went to a weaker Kozak sequence, he found that, in fact, the upstream ORF was more weakly translated, whereas the downstream ORF was more strongly translated. So by making such mutations, he could change the ratio of production of the upstream versus the downstream ORF from the starting point, which was a 1-to-1 ratio, to 3-to-1 or 1/2-to-1. So this gave us, at least a 6-fold range in terms of the ratio of production of the two open reading frames, which potentially is quite useful in synthetic biology applications. So the next question was, okay, those were endogenous proteins being produced. Can we now replace the open reading frames with any protein of interest to us? So in this case, we replaced, I just ask you to focus your attention on lane 2, where we replaced one of the open reading frames with the Venus reporter, essentially a GFP, and then the other open reading frame was replaced with an antibiotic selective antibiotic selection marker, emetine resistance, confirmed by ribosomal protein S14. And if you just look at lane 2 here, you can see, in fact, that in vitro, we could synthesize reporter and selective markers in vitro using the sequence context of the polycistronic messenger RNA. So all of this was very useful. And as I mentioned in the introduction, that Jeff Mosley had just joined the group, and he brought to us the Oxygenchlorella system. So again, this is a green alga that has been very popularly used in the biotech sector. And what Jeff pointed out to us, that here are 2 polycistronic mRNAs from Chlamydomonas and Chromochlorus shown here. You can see these 2 open reading frames. And he noticed that in Oxygenchlorella as well, there was most likely these 2 open reading frames were translated from a polycistronic messenger RNA, because we noticed that they were adjacent to each other in the genome, just as they were in many other algae. So Jeff replaced the upstream open reading frame. The 2 happened to code for mitochondrial proteins. He replaced the upstream one with the Thi-C, which is used as a selectable marker because it allows Oxygenchlorella, which normally requires thiamine supplementation for growth, as shown here. And then he replaced the downstream one with the gene coding for the enzyme beta ketolase. And the idea here then was to test whether we could express 2 proteins from a single transcript in the Oxygenchlorella system. And as you can see here, he selected for the upstream open reading frame. That is, he selected for growth in the absence of thiamine, as shown here. And then he tested we're showing you 4 random transformants here. And you can see he tested the 4 random transformants for expression of beta ketolase, which allows the precursor carotenoid to be converted to astaxanthin. So the cells show up red here. So this confirmed that, in fact, we can use this system for manipulating genes in vivo. And then on the last slide, what Jeff did was he again used 2 reporters. In this case, we're using sucrose invertase along with beta ketolase. And the reason we switched to these markers is that we can actually quantify expression from each of these. And he tested 2 different promoters, HUP1 and PGI1. In this case, the HUP1 promoter, we get very strong invertase activity, which allows the cells to grow on sucrose. So normally the organism cannot use sucrose because it doesn't have its own invertase, but we can introduce a secreted invertase. And now you can see the transformants can all grow. We notice a difference between the 2 constructs. In one case, the cells are green now because they've used up all of the sugar. So they're now relying on photosynthesis and rebuild a photosynthetic apparatus. So you can see that depending on whether we have one promoter or the other, we can actually get differences in gene expression from these proteins expressed in vivo. So we're quite excited about this system, and we're hoping to be able to use it to generate bioproducts in the algal experimental system. So I'd like to acknowledge now my coworkers and collaborators. I've mentioned that most of the project was led by Sean Gallaher and Jeff Mosley in the lab, both of whom have affiliate appointments now at EGSB. And we had excellent collaborative interactions with Chris Niyogi and Melissa Roth on the chromochlorus system. A lot of data from ficus, CSPs, and MZEL through JGI and PNNL. The in vitro translation reactions were done by Inian, graduate student with Steve Theg, and then Kristen Blavey and her coworker helped us to look at sort of the evolution and distribution of these polycystronically encoded proteins in various algal genomes. So thank you for your attention, and I hope we have time for a few questions.
All right, thanks so much, Sabeeha. That was a fascinating talk. So far, we have one question in the Q&A, which I had a similar question. This is from Alan Quo, who says, wow, what is the function of the polycistronic orfs? Are those functions related to each other? And are they organellar in origin?
That's a good question. We assume that many of them are probably organellar in origin, but probably like a billion years ago from the original endosymbiont. And this is something that Kristen has helped us with in terms of trying to figure out whether the functions of the two proteins are related. Well, first of all, as Alan knows quite well, many of the algal proteins, we don't actually know what the functions are. So for a lot of them, we cannot even address the question, but in a few cases, Kristen was able to document that they do participate in related function. The first one that was identified actually already maybe even more than a decade ago by John Pringle's lab at Stanford was the DNA repair protein REX1, where two subunits are actually encoded on a dicistronic mRNA. But I think Kristen identified a helicase and a ribonuclease that are co-expressed, presumably involved in RNA degradation. There's proteins involved in cell division and replication that are co-expressed, proteins involved in recombination, which again are co-expressed. And very interestingly, there are proteins that have something called a Lyre motif or LYR motif, and that's involved in mitochondrial assembly. And I think she found at least 4 such proteins encoded by polycistronic mRNA. So that was quite interesting. So the short answer is yes, they do seem to be related, but not, we haven't been able to document it for all of them.
Okay. And then, a follow-up question just asks, are they similar or the same genes between the different algae? Do you see the same genes as polycistronic?
Yeah, so I think about, and if Sean is in the audience, he may be able to correct me, but I think that when we looked at, so first of all, we only had isoseq data from Chlamydomonas, chromochlorus, and Dunaliella. So the other algae that we've looked at, we just have to sort of deduce that maybe they're polycistronic when we see them side by side, adjacent genes. So based on that, looking at like 5 algal genomes, I think Sean found that maybe about 30% of them appeared to be conserved polycistronic across all these algae. And now remember, we're talking about, 500 million years of evolution in some cases, or even maybe 600 years of evolution. So finding them conserved like that after 600 million years of divergence, we think is significant.
Yeah, absolutely. Well, a related follow-up question from me, these isoseq data are being generated for, a variety of species, but they're certainly not comprehensive for all the genomes that we know. Do you think that polycistronic gene expression is going to turn out to be much more common in in other types of organisms, or do you think it's algae have something?
No, I think actually, yeah, I think it is going to be common in a lot of organisms. As I mentioned, it was noted in a genome paper in cotton. And I think Igor noted it in his paper on mushroom-forming fungi as well. I think there was not as much analysis of those. They were perhaps thought of as some sort of oddity or something. And then in Drosophila, also there was sort of a substantial analysis showing hundreds of polycistronic mRNAs there as well.
Yeah.
With the very first one noticed in Drosophila, I think in 1998, for some sort of heat shock response, I believe. So we just haven't had the technology previously to find these things, A. And then B, I think the algorithms need to be modified as well, because typically there's been a preference to use an algorithm that looks for the longest open reading frame in a transcript. And if you noticed, many of the ones I showed you were only like 10, 12 kilodaltons. So they get missed as being part of the UTR or something. So I think we need to change that. And finally, the fact that we could translate in vitro in both reticulysate and wheat germ extract tells us that there is that capability to translate polycistronic mRNAs in quite divergent species. So I think that this will, the textbooks will be changing soon.
Yeah, well, and in prokaryotes, that being on a common transcript has been a strong functional signal for discerning functions, and so you might also might actually be useful for annotating some of these exotic genomes.
Absolutely, absolutely, yeah.
All right, well, I think we still have time. There's another question in the chat from Yan Liang that says, "Thank you for the great talk. Is there an internal ribosome entry site before the second ORF? Based on the already identified polycistronic genes, is it possible to predict other polycistronic genes in the genome?"
Yes, exactly. So that is something that we did consider, and Sean actually did look at the genome for that. And the short answer is no, we didn't find any internal ribosome entry sequences or any evidence that there were such things. We also don't think it's a 2A element type of mechanism, which is something that's being used a lot in other systems for synthetic biology applications or for ectopic expression of proteins. And we think that it's probably just leaky ribosome scanning. And the mutations at the Kozak sequence support that.
Okay,